to today's bite-sized bioinformatics, which will be about functional chromatin annotations. So this time last year, we actually had a Christmas special for bioinformatics and biscuits where we were decorating biscuits, but obviously that's not on this year, but I still thought I could at least have a snowy title slide. But also I was going to use this title slide or this Christmassy theme to start with an analogy, and you can tell me afterwards whether it works or not. But I think pretty much everyone, even before I started talking about Christmas, had the annotation of this picture, the idea that this was, with all its snow and the cold and the darkness and the Christmas tree, of course, that this was something to do with Christmas. And part of this is weather related, and we've actually got quite a lot of weather data for Christmas Day. So we've got nearly 200 years of weather data here, and we can see, a lot of information about temperatures and snowfall and humidity and all this. And if I look at this, there were only 14 days with snow over the years, but hey, we still associate this with Christmas. Now, we don't have this data only for Christmas Day, but we actually have it for every day of the year. So I'll continue with the analogy. We've got a calendar, our whole genome here, and we've got a lot of data for every single day for the last 200 years. So this is surely a lot of data. And because we can't look at it all individually, we will have to do something with it to make it accessible. And the first thing we can do with it is we can group it. We can take averages over the years, but we can also take averages horizontally, so to speak. So we can divide the whole year into segments, into months. There isn't a particular reason why we chose 12 months, really, but we can average across, and we get average temperatures, which you can see here, and you can get a rainfall graph, which you can see here. We can go even further with our grouping and go to seasons and say, for example, we say December, January and February is winter, and June, July is summer, and so on. We can also have some functional associations with this. For example, when we book our holidays. So it might make sense to book a skiing holiday in winter, as it's probably a lot more likely that we'll get snow in February rather than in July. Taking this one step further, it's quite obvious that temperature and rainfall profiles are very different across different regions in the world. Also, we all know that individual days might not be representative for the weather data of a month or even a season. So we all know about the cold day in July. And also, coming back to the initial slide, that when we look at the actual data, Christmas weather is much more likely to look like this, but we've still annotated Christmas with this, and so our annotation might not be accurate. Moving on to genomes, though, and genome annotations. We've now had many genomes for many organisms, and in the last years, we've also accumulated a lot of chromatin data across different organisms and different genomes. So you can chip different histone modifications and you get these profiles. You can complement these with individual transcription factors or things like DNA methylation, ChIP-seq, or polymerase-seq, or things like that. So you get genome-wide data for quite a lot of factors. And this, again, like the weather data, is a lot of data, and we somehow need to make sense of it. So people realized quite early on that some chromatin modifications tend to go together, like the ones here at the bottom. So these are different histone modifications that are plotted here, whereas others seem to be mutually exclusive. So people were saying, can we use all the information that we have in the histone data and sort of flatten it out to have an annotation that's just one per genome and per cell type? So can we sort of squeeze the chromatin information into a state information? Is the chromatin in a certain state? And here we see chromatin modifications for forebrain and corresponding annotations, chromatin states. And if we compare this to a different cell type, we can see that the chromatin profiles are different, but also the annotation tracks, so the states become different. So we've actually summarized quite a lot of information in this one annotation that comes from different chromatin modifications. You might have seen one of those heat maps, which is produced by one of those tools that do these chromatin segmentations, ChromHMM. And you would plot the marks that you've looked at, the marks that you've included in your segmentation on the x-axis, and here on the y-axis, you would see the number of states that have resulted. And the color indicates how likely it is that you will find a specific mark in a specific state. And then people take it one step further and give names to the states. And here we've got a number of different states, enhancers, promoters, bivalent regions, and they also usually get associated with a color. Hence, people talk about chromatin states, but they also talk about chromatin colors. There are quite a number of chromatin segmentation tools, and I've named some of them here in the middle, but this is by no means a complete list. And those tools actually do relatively different things. For once, they use really different models to go from chromatin modifications to chromatin states. So this is one of the major differences. But they're also different in the way they treat the input data. For example, you can have tools which use binary data. So either your mark is present in a certain region or it is not present. Or you can have tools which actually use a quantitative signal for each region. A lot of them have fixed windows, but some also use data-driven windows. With some of them, you need to tell them how many states you want at the end. And with some of them, they will also take this from the data and use a data-derived number of states. They're also quite vastly different in the amount of compute power that they need to run. When you read through all the publications, there's clearly one commonality, and that is that every single one is better than all the others. I don't want to enter this discussion here, but what I can say is that for each of them, you have two options of using them. So either you can run those chromatin segmentation tools yourself. You can, maybe you've produced a lot of data, or you have access to publicly available data, and you want to combine it to make a chromatin annotation. If you do that, you should really think carefully about what tool you use and which settings for those tools you want to use. And but this is really quite a non-trivial exercise, so I don't want to go into this for today. What's somewhat easier to use and to access is if you just use published state annotation tracks. And there are really quite a number available now, but because mainly because many of the big consortia, or some of the big consortia, have adopted ChromHMM as their tool of choice, and it's also been one of the very early ones, most likely you'll come across ChromHMM annotation tracks. So if something's published, there's a high chance that it will be from ChromHMM. There's a growing number of segue annotations as well, but really, since ChromHMM is dominating the market, if you wish, at the moment, I'm going to go into this in a bit more detail. So when you run ChromHMM, you actually need to make a few key decisions on how you want to run the program. Window size is probably the easiest. It defaults to 200 base pairs, and literally all the annotation tracks that I have seen so far keep that window size, so that's probably not too much to worry about. other than that this will be your resolution. Then next up is you have to decide what chromatin marks you want to include. So this is from the original ChromHMM publication. You can see that they've had an awful lot of chromatin marks. And it's quite obvious that the states can only reflect the chromatin marks that underlie them. And it's also clear that just when you look at the profiles, if you take one of these out, your states probably aren't going to change that much. But if you're going to take something like CTCF out, which is very different to the other marks, you will either incorporate this in your state annotation or not. So very often, of course, your annotation will be limited by the data that's available, but clearly this has a big influence on what your annotation track will look like. The next thing you have to decide on is how to binarize the signal. And this is sort of one of my favorites because it's not straightforward. So ChromHMM uses a binary data input model. So every 200 base per region is either labeled as marked or labeled as unmarked. And splitting chip signal into regions that are marked and unmarked is a common problem. For example, if you want to call peaks, and it's not a straightforward thing to do. If you look at those two modifications, so this is real ChIP-seq data for K27 acetylation or K27 trimethylation, you can see that there are very clearly regions, for example, this one here, where you've got a high signal for K27 acetylation. There are also regions where it's pretty clear that there is a very low signal here, in this case for K27 trimethylation. However, there's also an awful lot in between. And where you set the threshold will influence what your annotation will look like afterwards. Also, the different levels might actually be biologically relevant. So a highly marked region might have a different function from a lowly marked region. ChromHMM itself, the software, does the binarization based on a Poisson background model. But because the authors acknowledge that there are different ways of doing this, you can also do the binarization yourself and put a different threshold in. You also have to tell ChromHMM how many output states you have. So here we have a small model for only 4 chromatin marks, and these, in this case, define 8 states, and you get different probabilities for, say, the K4 monomethylation mark being present in state 1. You can go down the states. Exactly the same data can also be used in ChromHMM, but you tell it to do up to 16 states, and you can clearly see that states come up with different combinations, and the resulting models will be quite different for this. You also have to tell ChromHMM in which mode it should run. So if you've only got one cell type, you can use the marks that you've got and make a model for it. And you will get the resulting states with the probabilities for each mark. If you want to look at a second cell type, you could do the same thing again. However, it will result in a different model, and then state 1 of the first model will not be equivalent to state 1 or any of the other states in the second model. So this doesn't work if you want to compare the chromatin states across more than one cell type. So for this, you need to use the concatenated mode, and that works by combining all the data for all the cell types, putting them all in one model, and then you end up with a situation where state 1 in cell type 1 is the same as state 1 in cell type 2. Also, you've got to decide on the annotation. So you've got to give the states names, because all you get out is a state and a number from the ChromHMM. In theory, you can give it whatever name you want, and these are more unusual names for the chromatin states, whereas here, this bit more conventional. So for example, a state where you've got K4 trimethylation very likely, or in K27 acetylation very likely, you might call active promoter. or one where you've got a high probability of K27 acetylation and K4 monomethylation, you can call active enhancer. So a lot of people will agree with that. But then if you, for example, look at this state number 1, where you've got K4 monomethylation, but really none of the other marks, you get a whole set of names for it. They might be called naive, intermediate, primed, poised, or inactive enhancers. And also remember that the states from different models aren't necessarily equivalent. So naming is another thing that can be very different between different studies. This here is data from ENCODE, because ENCODE also has quite a few chromatin, ChromHMM annotation tracks. And here are the histone modifications and factors that ENCODE has monitored, and this is ChromHMM with the respective probabilities. And here you can see the list of names, what they've called their states. If you're using something like this, say from the ENCODE project, then at least you've got some big names to go with it. But that doesn't mean that individual states from other publications are any less relevant. Again, coming back to this, just this list of ChromHMM key decisions. Talking about these, you will have realized that these will have an impact on the annotation that results. And getting this wrong might easily result in an annotation that's completely useless. And remember that when you're using a published annotation track, that someone will have made all these decisions for you. So if you're using published chromatin annotations, then you really need to treat them with care. Some of these decisions, there isn't really complete right or complete wrongs, but there is really room for interpretation. And someone has done this interpretation, and it could have been completely different if another person had done it. Also be aware that the annotations with the same name don't necessarily mean the same thing. So what someone calls an enhancer or a strong enhancer, because this is a most frequently disputed thing, might not be the same that someone else uses in a different publication. Very importantly, make sure that you're using an annotation that matches the cell type that you're analyzing. I recently came across an example where people have used a stem cell annotation for adult tissue. Because chromatin marks are very different between different cell types, of course, the resulting annotations are as well. So make sure it matches. And if you want to do anything with it in depth, then you really need to use the actual ChIP data rather than just the annotation. Where do you get these annotation tracks? You can download them, for example, from UCSC. So the Genome browser offers ChromHMM annotation tracks, and there's also a considerable number of segue annotations from different species. You can also find a lot of them on the Roadmap Epigenomics website and the linked WashU browser. So this is a very good source if you're looking for human. Then a lot of the annotations, especially from other tools apart from ChromHMM, you find in individual publications. They are either published as supplemental data or in one of the repositories. And what can you use them for? Well, so originally they are mostly developed to compare epigenomes across conditions, especially for the big projects like the Roadmap project. And but if you're doing this, you're probably quite likely to have your own data that you want to compare for your specific conditions, for your specific cell types. I've recently seen that there are also tools where you can systematically compare annotation tracks, for example, EpiAligner, but I haven't looked into this, so I'm keen to discuss if anyone has more experience there. You can use those annotations as context for other large data sets. For example, Hi-C, when you say, okay, the fragments that interact are usually of a particular chromatin state, that's one thing. Or another thing which is very commonly used is to give some context to any sort of region of interest that you're looking at. Very often these are ChIP-seq peaks. We just ask, well, the peaks are predominantly in which genomic element in terms of its chromatin marks. And because I've had several requests for this from different projects, I would like to quickly introduce to you to the Shiny project that I've started. This is probably currently only for the Vibram people, and it's really only a skeleton version at the moment. It only uses mouse ESL ChromHMM annotations. But what you can do is you can upload any set of regions that you're interested in, and we will give you two graphs, one just saying in how many % fall into which of these chromatin states. And it will also give you some enrichment of this state over its distribution in the genome, and you can download these data. So I'm quite keen to explore this further. So if anyone's interested and wants other annotations or other features, please just get in touch. Okay, and I would quite like to end here on this questionable annotation of our Christmas weather, and I'm very happy to take any questions. Oh, okay. Well, you can contact me anytime if you want to discuss any of this further. And yeah, if there aren't any questions.
Crystal, sorry, I can ask one.
Of course, yes, please. Sorry.
Hi, this is Courtney. I was wondering if you could use such a software to determine whether the distribution of a mark is atypical in your cell type, for example. Like if there'd be any way to compare with sort of existing data sets to say whether it's proportionately more or less or at different regions of the genome than you would expect, given the looking at a number of other cell types, for example.
I guess the answer to that is yes and no. In theory, this is precisely what the roadmap project, for example, is doing. They're profiling lots of different tissues with the same chromatin marks and use the same model for those, because you need to keep a lot of the parameters the same to actually be able to compare them, starting from the obvious chromatin marks that are in the model to actually using the same model. So yes, it's absolutely doable. If you just have one cell type that you want to profile for, or you've got some ChIP data for it, it's really hard to put it into context with other annotations because they're likely quite different.
Right. And probably dependent on like coverage and IP efficiency.
Yeah, I mean, yeah, all these things come in as well, but it's probably even more substantial than that because you need to be in the same model to be able to compare across the different cell types. I can see a question from Chris in the chat. Are there any plans to add human ESCs to the Shiny app? If you want them, just let me know. I can certainly do that.
Thank you, Crystal.
Okay.
If there's no more questions, then we'll end it there. And as Crystal said, she's very happy to receive any questions if people have them later on as well. So thanks very much for that.
All right. Thanks, everyone.